A child born in 1953. The structure of DNA has just been discovered. 1989, and this baby's genetic fingerprint can be identified. The first single gene for Huntington's disease has been discovered. 2003, this child's entire genetic code can now be read, and faulty genes in his DNA can be adjusted. Another birth, but this time no ordinary miracle. The baby's sex and eye colour were decided before she was conceived. Also her hair, the shape of her nose, and her intelligence. The date of her birth? Perhaps only a few years from now. She's born of a revolution in genetics, a revolution where each new step brings new questions of ethics and responsibility. And as the promise of the science gets greater, so the questions for all of us get bigger. The 26th of December 2002, a press conference in the USA.
I'm very, very pleased to announce that the first baby clone is born.
This claim by a religious cult, the Raelians, is almost certainly a hoax. But a cloned baby is the very real aim of some scientists around the world. It's the ultimate goal in a science where some fear the pace of developments may be outstripping our ability to examine, consider, and weigh up their consequences.
There are 72,000 experiments going on in the world today dealing with DNA. 72,000 experiments, and we've just begun. What we're doing is we're short-circuiting millions of years of trial and error and speeding up the process of gene transfer in a short moment. So I believe, rationally, I would have to say and go on record, I think it's insane.
Insane or not, it's the amazing idea that we can manipulate and control our genetic inheritance that drives modern medicine, as it attempts to outwit the major diseases caused by our genes.
When it comes to medical research, any medical technology that works is very quickly accepted by the public. Ethicists may not like it, but scientists may not like it. But the public, if they believe it works, will accept it. And legislation will always follow. Ethics has always followed science. It's never led it. And I don't see any reason why genetics is going to be any different. Ethicists would love to tell geneticists what to do, but I'm afraid the geneticists aren't going to listen.
As the debate rages on, this program will take you to the frontiers of genetic research. It's a place where scientists admit that often they don't know why their experiments work, but where there's no doubt that genetic medicine is showing extraordinary promise.
In the future, we're hoping we can inject stem cells into a patient, and those stem cells will repair worn-out body parts.
I think cloning is probably the most powerful technology that humans ever developed.
Our hope is that the work that we and other scientists are doing will one day result in consigning genetic diseases to the history book.
To find out what the public thinks about the developments that are revolutionizing our genetic future, Discovery commissioned the unique international poll across 8 countries. Our poll found that many people share the overwhelming optimism of scientists that genetics will decide the future of the human race. Most support was found in Brazil, least in the UK and US, but even here, 2 out of 5 people agree.
As we go on, I think it'll eventually, really influence our medicine and the way we treat people.
I think genetic developments will be just the future of medicine. I think a lot of ailments are the result of mutations in certain genes or deletions, and if we can address that, we'll just open, doors all over the place. I mean, it'll be a great advancement, and I think that's where everything is headed, is to gene therapy if it can be perfected.
I think there's a potential for great improvement with genetics. I think there's a lot of diseases that can be helped greatly within a short time.
I think there's a lot of hype involved in it, and there are other things that are going to probably do more to form what's going on than just genetics.
Much is promised by genetic science, the manipulation of our genes. But can it deliver? And if it does, are we ready to take on the responsibility of meddling with the very fabric of life itself? our DNA. London, in 50 years' time. We've imagined a world where genetic science is a part of everyday life for families like this. For this generation, genes can be chopped and changed at will. Designer babies are the norm. And a cure for the last great killer disease of the Western world has arrived at last. Animal genes are manipulated to suit human needs and appetites.
Unhappy being bald? Losing out with the ladies? Have the Maggie Hair 3 gene implanted into your scalp now.
Am I going to need that gene?
A genetic cure for baldness is some way off. But from the moment the secrets of our genome were discovered, scientists wondered if they could manipulate our genes. This big idea is gene therapy. For diseases caused by single gene defects, like cystic fibrosis, muscular dystrophy, or haemophilia, gene therapy seems like the ultimate promise. This man is living proof that gene therapy can work. He is the only person in the world to have been apparently cured of the devastating genetic disease haemophilia after taking part in a pioneering gene therapy trial. Diseases such as haemophilia are caused by malfunctioning genes. The idea behind gene therapy is simple. Why not insert a fully working gene into the DNA to do the job properly? Gene therapy uses a virus to insert the new gene into the nucleus, much as an envelope carries a letter. The gene randomly inserts itself into the genome. Now it can be read, and correct instructions passed out of the nucleus to the protein-making factories, which in turn produce the building blocks of our bodies. But gene therapy has its risks. Indeed, in some trials, it's proved fatal. In 1999, teenager Jesse Gelsinger died while undergoing experimental gene therapy for the liver complaint OTC. The virus being used to carry the replacement gene triggered a huge immune response that killed him.
My problem with gene therapy is not in the principle of it, but its execution. The young Jesse Gelsinger, this young man was not near death at all. He was leading a fairly healthy life. He died almost immediately from the actual therapy, so he died and he didn't need to.
Other mistakes have arisen from gene therapy. Children with immunodeficiency disease who are forced to live in a germ-free environment seemed like ideal candidates for the treatment. That was until two French boys developed leukemia when the randomly inserted gene designed to cure the condition disrupted a neighboring gene called LMO2. This caused blood cells to divide uncontrollably, resulting in the cancer. Some doctors believe the promise of gene therapy is so great that they're pressing ahead and trying to eliminate its risks. This is what they're doing in a joint trial between the Children's Hospital of Philadelphia and Stanford University for the single gene disorder haemophilia.
As the blood gets older, it'll get darker, but what's concerning me is that it's actually bigger than the other side, so it's not just a bruise on the skin.
2-year-old haemophiliac Dylan has a faulty gene passed on from his mother on the X chromosome. His body doesn't produce enough of the blood clotting agent factor 9, so he bleeds very easily. Today he's receiving an emergency dose of synthetic factor 9. The factor is expensive, between $50,000 and $100,000 per patient per year, so over half the sufferers in the world can't afford it. Patients can become immune to it, and it's painful to administer.
Jack, you know what? Actually,
Look, I'm going to right here, arm down. If you want to hold his right arm. Good boy.
Okay,
For 6 years, Dr. Katie Mano and Professor Kathy High have been working to design a safer virus to deliver a synthetic clotting gene.
Should we take it out now?
We'll take it out.
Please.
Hemophiliacs like Dylan only produce around 1% of normal clotting factor levels. Just a slight increase would make all the difference.
People who have slightly higher levels, say 3 to 4%, those people experience many fewer spontaneous bleeding episodes or dangerous bleeding episodes. So if we were able to maintain circulating levels in the range of a few percent and would allow him to basically be free of the fear of spontaneous bleeding episodes.
4 months ago, 31-year-old Chris Brent travelled from Australia to the US for the trial. He and his doctor had spent months persuading the Australian government to allow him to take part.
It's oftentimes quite difficult to know what a lot of these regulatory committees require, and so it takes a lot of time to fill out the paperwork, make certain that all of the right things have been done. There's no doubt that the tragic death of Jesse Gelsinger has changed not only clinical gene therapy trials, but in fact all clinical trials the world over.
I was aware that studies in America on gene therapy had been halted in 1999 after there'd been a death in a trial of someone who had OTC deficiency. There were some small concerns, but I was probably more interested in the big picture that could potentially benefit a lot of people.
The synthesized factor IX gene was injected directly into Chrissy's liver, a simple one-off procedure. Within 3 weeks of the trial, Chrissy's body began to produce its own factor IX, something he'd never experienced before. After 6 weeks, the new gene had pushed his factor level up to an amazing 12%.
I mean, when I found out that my body was producing its own factor IX, it was quite exciting. You know, I felt probably that I could push myself a bit further and wouldn't have to worry about any bleeds.
This is the goal that we had been working for more than 5 years, nearly 10 years. So it was a really wonderful moment. We have learned through the years not to get too excited until time goes by, but we were cautiously optimistic.
Yep.
And Professor High was right to be cautious. After the initial improvement, Chrissy's factor IX levels began to drop back down. In addition, his liver had an immune reaction to the new gene, causing hepatitis.
I guess I was a bit disappointed that, this factor that I had started to produce was now dropping, but other than that, I don't think it caused me any great anxiety. My liver's fine now, I believe. It certainly can cope with a few drinks, so that's all I ask of it.
Our poll asked people if they'd carry on with gene therapy if they knew patients had died from the treatment. Very few from the UK or America would continue.
I think it would depend on my circumstance. If it were, if I were terminally ill, if this was something that could kill me, I don't think there would be many choices left, and I would be for it.
If I was dying, I would probably still be willing to take a risk, but I suppose you would always have to weigh it against side effects and how long you were likely to have your life prolonged.
If someone had died from having the treatment before they wanted to give it to me, I would hope that it had been through more trials.
I feel very bad about messing with our genes. Of course, our genes have to develop in a natural way. I don't think we have to manipulate with them.
It's an unfortunate truth in medicine that many treatments which become standard in their early days were tremendously dangerous. The classic one is of heart transplants, and the early heart transplants in the 60s had no chance of survival at all. And they wouldn't be allowed at all nowadays. But if they hadn't been done, we wouldn't have the standard technology of heart transplant. Now, gene therapy today is nothing like as risky as that. But it would be foolish to deny that there are real risks, some of which perhaps we don't know yet. However, if we don't take those risks, gene therapy will never go anywhere. So rather reluctantly, I think we do have to take them, even though it may cost, unfortunately, some human lives.
For Chris, gene therapy is a risk that appears to have paid off in an unforeseen way.
Well, since I had the gene therapy, which was 4.5 months ago, I've only had one bleed. So that's pretty extraordinary for me. I normally would have one bleed every 2 weeks. And it's actually been 3 months now since I've had a bleed. Can't entirely explain why, but it's been very good.
In Australia, there are strong hopes that Chris Brent's experience will allow more gene therapy trials to go ahead.
The consequence of this is very exciting and indeed is a major advance in gene therapy insofar as we're seeing the first glimpse of the possibility of a therapeutic benefit in a human being receiving gene therapy. In addition, he still seems to have been benefiting from the gene therapeutic procedure in a way that we don't fully understand.
Even with successes like Chris, some believe we're trying to run before we can walk. We just don't know enough about our genes to start experimenting with them to this extent.
We only have 30,000 or so genes, but there may be 200,000 or 300,000 proteins those genes code for. What the molecular biologist can't tell you is why does a gene decide to code for one protein versus another? So I think what we have yet to explore are what are those subtle factors out in the environment that may affect why genes become one protein versus another, and that's way down the line.
Replacement gene therapy involving the introduction of new genes will continue to carry risks until we know exactly how genes work. But new research here at the University of York could prove much safer. Instead of replacing a faulty gene, it destroys its message by attacking the messenger. It's the first step in tackling the biggest killer of all, cancer. This is the nucleus of a cervical cancer cell that has been forced to self-destruct. The cancer is caused by a sexually transmitted virus called HPV. Every year, 400,000 women across the world are killed by it. Medical pioneer Professor Joe Milner has spent 3 years trying to find ways of defeating it. Inside the nucleus of a healthy cell are the genes P53 and RB. These two genes suppress tumor formation by controlling the rate that our cells divide and destroying any damaged cells that might become cancerous. Cervical cancer occurs when the cancer virus enters the cell and attacks the tumor suppressor genes by placing two of its own genes, E6 and E7, into the human genome. These take over the reins of cell growth and force the cells to divide in an uncontrollable manner, causing cancer. Professor Milner's study, at the cutting edge of genetic research, is called RNA interference. It treats the cancer at its genetic root.
Conventional treatments of cancer, including chemotherapy and radiotherapy, are effective for various tumour types, but have the disadvantage in that they also damage normal cells. In other words, that they're non-selective for the cancer. The huge advantage of RNA interference is that it can be tailored to selectively target cancer cells.
Genes are read by messenger RNA, which takes their instructions to the protein-making factories in the cell. Professor Milner's technique prevents the message from the cancer genes reaching these factories. It stops the message by effectively shooting the messenger. The agent that does this, a molecule called short-interfering RNA, is introduced into the cell that's been infected by the cancer virus. Here it combines with the messenger RNA, carrying the virus's cancer-making instructions to the protein factory, and destroys it, causing the cancer cells to self-destruct. On paper, it's an amazing concept. The question was, could Professor Milner get it to work in practice on these killers, the cervical cancer cells?
You can see that the picture immediately is different. There are far fewer cells, and here the cells with halos around them. These are cells which are in the process of committing suicide, essentially. These are dead. These are the remnants of cells, of the cancer cells. It was a wonderful feeling looking down the microscope and seeing these results. You could see that those cells were dying, whereas normal cells, which had had exactly the same treatment, were completely unaffected.
Now we've got a technology which allows us to interfere directly with the actual RNA genes of the virus. And certainly in its early days, the progress has been quite remarkable. So what we might have is a new generation of medicines.
From Professor Milner's research, you might think a cure for cervical cancer was imminent, but human trials are at least 3 years away. Our poll has found overwhelming optimism that genetic science will bring cures for most diseases. The British and Americans are more sceptical, but even here, more than 3 out of 5 people believe genetics will cure disease.
I think genetic manipulation, not at conception, but past that, is going to be the biggest course in medicine probably in the next 20 years.
Hopefully the developments in genetics will continue to improve our lives.
Gene therapy and research like Professor Milner's offers hope to an end to many genetic disorders where only 1 or 2 genes are involved. But in other big genetic killers like heart disease, a complex combination of genes is at work, and a different approach is needed. Radical techniques using the very latest genetic research hold great promise, but they bring a whole new set of ethical questions. A year and a half ago, Nelson Aguirre was knocking on death's door. He had chronic heart disease after several heart attacks. Without a transplant, his prospects looked grim.
I've had 2 heart operations, including 7 bypass procedures. I couldn't climb the stairs, and I had a lot of chest pains. I had no will to live. I was a man that wouldn't survive a 3rd heart attack.
But instead of a heart transplant, Nelson underwent a procedure that has brought about an amazing recovery. His severely damaged heart was effectively regenerated, thanks to one of the most controversial areas in genetics today, stem cell therapy. Stem cells are the master cells of our bodies, regenerating and repairing different tissue types. In the past, scientists believed the fate of adult stem cells couldn't be changed. They could only become what they were destined to become. But now, scientists are exploring the idea of taking stem cells from one part of the body and tricking them into becoming cells from another. One organ with almost no stem cells of its own is the heart. Here, dead or damaged heart tissue cannot regenerate. But could stem cells from another part of the body be fooled into becoming new heart cells?
The notion that you can retrain your own cells to replace, for example, damaged tissue is a tremendously exciting one. It really is. I would say it's the most exciting area in modern genetics, or cell biology, which is what it really is.
What switches genes on or off to instruct a bone marrow stem cell to become, say, a white blood cell is still very uncertain. But remarkable work being carried out here at the Texas Heart Institute could hold some of the answers. Dr. James Willison, who initiated the research, believes adult stem cells could be far more powerful than had previously been thought.
We've become convinced that one can use bone marrow cells as a form of stem cell transplantation in a way that will improve blood flow to the heart and improve the function of the failing heart.
Stem cells in Nelson's bone marrow are sucked out using a syringe and filtered to separate off other cells. They are then injected into the ailing heart, between the living and dead tissue. The big question was whether this new environment could persuade the stem cells to change into new heart cells rather than the white blood cells they were originally destined to become. Dr. Emerson Perrin, a heart specialist at the Texas Heart Institute, began to build upon the pioneering work. He decided to conduct the trials in his native Brazil. Dr. Perrin has to map the diseased heart in 3 dimensions by putting a catheter into the organ via an artery in the leg. As this map builds up, it represents Nelson's heart before the stem cell injections. In a healthy person, this would all be purple. The red areas show where his heart has been savaged by heart attacks.
Here you can see a purple spot. This area contracts normally. So you can tell immediately that this heart is very, very sick. If we lift this heart up and look at the bottom surface, these black dots are the actual sites in which we have injected the stem cells in the area most needing better blood supply, better blood flow, and where there was potential for recovery.
The next part of the 45-minute procedure involved precisely injecting the stem cells into the heart using a tiny syringe fed up into the beating organ via the artery in the leg. A three-dimensional map is then used to guide the injections to a fraction of a millimetre, hitting the predetermined areas needing most help. Although these stem cells are from the patient's bone marrow, once placed in his heart, something amazing has happened.
It turns out that when you put one of these stem cells in the middle of a bunch of cells that are all heart cells, they sort of mimic the environment. The cells are very smart in a way to turn into the kind of tissue that we need to turn them into. We think that from one standpoint, the stem cells are able to secrete many different very beneficial substances that may, for example, help grow blood vessels. They may also turn into, let's say, a piece of blood vessel or turn into heart muscle, and that's another mechanism. So at this point, we're really not sure exactly how that works, but we have a fair idea.
They may not know how it works, but it seems it does work. The image on the right is a map of Nelson's heart just 16 weeks after his operation.
So we've been looking at this map at the time of injection, and you can see here at the site of where the cells were injected, we have a marked improvement, which is demonstrated by this large purple area, which represents now normally contracting muscle. And if we look at the other side of the heart, we see also a marked change in this large red area of weak heart muscle now turning into a near normal, stronger beating heart.
So far, 14 patients have undergone the procedure, and all, like Nelson, have made remarkable progress.
Every other day I walk 4 kilometers and I don't feel anything. I swim in the ocean and I've now gone back to work. It's given me another life. I'm reborn.
The trial is still in the experimental stage. It's unclear how the genes are turned on and off in the stem cells, but doctors are pushing ahead with new trials to verify their data. Patching up the heart seems like a fantastic way to regenerate the organ. But what if we could bypass this stage and build a new heart in its entirety? That's the ultimate aim of stem cell research, to replace organs altogether. But to do so moves the debate into an area of huge controversy. Adult stem cells may be much more versatile than was previously thought, but still their use is limited. To attempt to build a new organ from scratch requires the most versatile cells of all, embryonic stem cells, cells whose genes can switch on and off to become all the cell types in the human body, cells derived from embryos around 5 days old.
The most exciting part of genetics, without question, is the genetics of development. How do you get from an egg to a human being? How do you get from a streak of tissues in the embryo to a heart? Now, if we can crack that, it's conceivable, only conceivable, that we can do the job ourselves and regrow a heart. I mean, think of the medical problems it would solve.
But to solve those problems requires a step that many find repugnant.
When we create embryos, specifically to harvest stem cells for experimental purposes, with the idea that then the embryo will be destroyed so it won't come to term, that is really dangerous. That's eugenics. That should not be the way we proceed with science.
But the science is proceeding, and with such extraordinary results, the ethicist's voice may fight to be heard. Growing new organs for transplant sounds like a Frankenstein fantasy, but it's becoming reality here in Japan. This frog has eyes that were grown from scratch artificially in this lab. They work perfectly after being transplanted into the sockets of the frog by this man. Professor Makoto Ashishima has made an amazing breakthrough. He's the first scientist in the world to create from scratch a sensory organ from an embryonic stem cell. Embryonic stem cells are the building blocks of all creatures, whether frog or human. After a sperm has fertilized the egg, it begins to divide until after 4 or 5 days it's a 200-strong ball of cells. These are stem cells. At this point they have the potential to become any part of the body. If we can harness this potential and direct it, we can grow any organ of our choosing. At the Tokyo University, Professor Asashima has taken stem cells from a tadpole embryo and cultivated them in the lab. The challenge is then to persuade the stem cells to turn into eyes.
We put various inducers into a test tube with the stem cells. By altering the condition of the cells, such as changing the concentration of a protein inducer called actin, we can create different organs in the test tube.
These eyes, grown in the lab, are transplanted into tadpoles who've had their own eyes removed. The next challenge is to get the transplanted eye to work. And once again, while genetics has mastered the mechanics, quite how the engine is switched on remains something of a mystery.
The nerve on the eye grows towards the optical part of the brain and then it stops. A nerve then comes from the brain and the two nerves connect. At the moment, we just don't know why the nerve from the transplanted eye grows towards the brain.
As the tadpoles mature, the million-dollar question is, does this pioneering science actually work? Can the young frogs see through their new eyes? To find out, Professor Asashima has harnessed an unusual natural quality of the frog.
If the frogs can't see, the body of the frog becomes dark. If the frogs can see, even through one eye, they become a much lighter colour, sometimes white.
In this tank, you can clearly recognize the lighter colour frogs that can see. Some are even white. And the darker colour frogs where the transplantation hasn't worked, and they are blind. Professor Asashima's hope is that this futuristic vision could one day quite literally give eyes to sightless children.
There are different degrees of blindness, but if we can grow eyes in a test tube, then there's a chance we can cure the blindness using stem cell treatment.
As Professor Asashima increases his knowledge of how the genes work that govern these stem cells, he is learning to grow other frog organs, kidneys, livers, and one of the most complex organs of all, the heart. This beating frog's heart is waiting to be transplanted. It's the next objective on the professor's list. In Professor Asashima's experiments, the stem cells are derived from embryos sexually produced by male and female frogs. The big problem for humans is that our complex immune systems instantly reject organs which contain DNA other than our own. This is why heart transplant patients have to remain on powerful immunosuppressant drugs. The only way to obtain stem cells containing purely our own genes is to make a cloned embryo. To achieve this, a human egg is stripped of its own nucleus and a new nucleus is inserted. It could come from any cell in the body. An electric shock fuses the two together and starts cell division. A cloned embryo is created. And Dr. Robert Lanza is one man doing just this. Here at ACT Biotech in Worcester, Massachusetts, they are attempting to bring an embryo to a stage where its stem cells can be harvested.
We've used the cloning technology to actually generate early-stage human embryos that are at the 4 to 8 cell stage, to go from the early stage embryo to a blastocyst, which is a cell cluster of about 100 cells. It requires fine-tuning the culture conditions as well as each step in the cloning procedure.
Robert Lanza is encouraged by his research with cloned embryos from our nearest relatives, the primates. Here they've reached a crucial blastocyst stage, 100 to 200 cells where the stem cells can be harvested. He believes the applications are wide-ranging.
We already know, for instance, how to turn embryonic stem cells into whole dishes full of dopamine-producing cells that could be used to treat, for instance, a patient with Parkinson's disease. By adding something known as retinoic acid, we can turn these embryonic stem cells into beating heart cells. We've also already seeded those heart cells onto various synthetic scaffolds to create what we call myocardial patches so that we can fix the heart very much like you would fix a bicycle tire.
Success with primates has encouraged Robert Lanza in his ambition to create whole human organs from cloned embryonic stem cells.
To create an entire heart, that's the holy grail. That's a very complicated procedure, and that's probably a good 10, 15 years off.
Although a while off, it all sounds very promising. But there's a price to pay. When an embryo is harvested for its stem cells, it is destroyed in the process. The end of a potential life. The issue of whether this tiny ball of cells has the same rights as the rest of us is proving so controversial, many people think the research should be banned. Our poll shows while in Brazil and Turkey, 2 out of 3 people believed it should be outlawed, in the USA, 2 out of 5 think this. In the UK, half think it should be banned. The right to life issues are so complex, the world is divided on whether stem cell research with cloned embryos should go forward. Great Britain still allows research into embryonic stem cells up to a 14-day limit. Health ministers in Australia can't reach a consensus on the issue. Singapore has appointed a panel of experts to study the ethical question of using embryonic stem cells, and there is no law at all regulating stem cell research in Israel. The United States has banned the use of taxpayers' money in embryonic stem cell research, and political pressure is building to ban it altogether.
Fortunately, it hasn't stopped there because stem cell research is so important that many private bodies are funding it and many European governments are funding it. So unless you've got a universal revulsion against some kind of research, it will tend to go on. And the stuff that works, you can rest assured the law will accommodate it without turning a hair.
And when that happens, the most difficult choice you may have is whether to order your new organs online or from some salesman who knocks at your door.
Good morning to you, sir. Can I have a moment of your time, please?
Not interested in anything you sell.
Sir, please. All I ask is 1 minute, and I can make you an offer your body cannot refuse. How old are you, sir, if you don't mind me asking?
40.
Do you know that when you're 40, your liver is not as efficient as it used to be when you were 18? And your heart is starting to fail too.
Yeah, so?
How about a new heart?
The therapeutic cloning research, which tries to grow human tissues with the hope of replacing, let's say, damaged heart tissue, could, I'm sure, be abused. But 20 years from now, the notion that you should not use stem cells because the cells were taken perhaps from an egg that was fertilized and not implanted in an IVF clinic will strike us as equally outrageous.
The whole concern comes down as to whether or not a microscopic ball of cells smaller than the head of a pin warrants the same rights and respect as an adult or a child who may die because we fail to use this technology.
The greatest question for lawmakers, ethicists and the rest of us is what happens when embryonic cloning is taken to its biological conclusion. Growing a cloned human embryo in the lab beyond a few cells is proving difficult, but growing one in the womb could be much easier. Scientists are learning from cloning work with mammals, but objectors point to Dolly the sheep as evidence that it should never be allowed to happen in humans. Professor Keith Campbell was Dolly's co-creator. He's listened to the argument that Dolly's arthritis and early death can be blamed on the fact that she was a clone.
To my knowledge, I don't think there's any evidence that Dolly aged prematurely compared to another animal of the same age which was produced by natural breeding techniques. Don't let it appear that I'm saying that all of the animals we produce by this technique are normal. It's a very inefficient technique, and we don't understand the mechanisms which control development. So we produce some abnormal and also dead fetuses and animals.
Despite the animal data, some scientists are forging ahead in pursuit of the ultimate aim, to clone a human. Critics ask why should anyone want to do this? One of the few compelling arguments is to give infertile couples a baby. One man actively trying to produce a human clone is Dr. Panos Savos.
We are at a very advanced stage as far as our human cloning program, and we are obviously quite confident that we can execute and we can create clone embryos that are healthy, can implant those clone embryos and yield a viable pregnancy, and I hope to deliver the baby at the end of this year.
Our poll asked when a human will be successfully cloned, and the most common belief is that it's already happened somewhere in secret. A third think it will happen within the next 5 years, while only 1 in 7 believe there will never be a successful human clone.
I think what we can assume here is that if it hasn't already been done, it's going to be done. We're going to wake up one day and someone will have done it. This is no longer a technical problem. This is just a question of which researchers are likely to pull it off first.
Good God, look at this. You in 2007. She's getting to look more and more like you every day. Seriously though, when we decided to get you cloned, well, for some reason I thought she might be a bit different to you.
There's no doubt that every couple should have the right to reproduce and choose the option via which they choose to reproduce. The people are so scared about cloning because they don't know enough about it.
Few scientists doubt that a human clone is already in the making, and that such a development is full of risk. Even those who have successfully cloned animals believe human cloning is a step too far.
The offspring may well have abnormalities which, with human medicine, may not be life-threatening, but I think that the chances of that happening are not worth the risks at present, and we should not attempt human cloning.
I think what people have got to realise is that if you develop human cloning, you're going to start experimenting on children, on babies, and there will be things that go wrong, there will be suffering caused, and it simply can't be justified, that kind of experimentation on people, because somebody feels that they want to perpetuate their genetic makeup.
Dr. Panos Savos believes fertility specialists like him are perfectly placed to avoid the risks.
We've been doing IVF in humans for the last 24 years, and therefore we know more about human reproductive medicine, how to create a child or an embryo in a dish, transfer it in utero, and then monitor that pregnancy and make sure that that pregnancy is guided properly to a very successful fruition. And we're very good at that. The bottom line is this, that cloning will eventually become just another mode of human reproductive technique that people would really go to as a last resort.
Our poll reveals intense opposition to cloning, even for infertile couples who cannot have a child any other way. Almost 9 out of 10 people polled in the UK said they wouldn't want to clone themselves even if they were infertile. The Danes are even more opposed to the idea, but a third of Mexicans support cloning.
We are going to be genetically modifying people, perfect specimens, you know.
I don't think there's a place for cloning at all.
Reminds me of my mother's curse. I hope your children are just like you. No.
Cloning myself? I think one Miles Miller in the world is enough to satisfy.
I'm with him. There's no way.
Yeah, I wouldn't want more of me running around. That would be scary.
Yes.
I would never consider cloning myself for me. That's not for me. I don't know if it's wrong morally, but I don't think I'd do that for myself. I think it'd be a little too weird for me.
Many of us, it seems, find human cloning abhorrent, but some believe that distaste among scientists will disappear once the first steps have been taken.
The scientific community, they're saying, oh, no, this is irresponsible, it's unethical to clone a baby. But then when you dig deeper, the scientists are really not opposed to cloning. They're just saying it's not safe yet. We haven't perfected it yet. They're really only opposed to those researchers who are jumping the gun because it isn't safe or because the scientific community hasn't put its blessing on it.
The only way to stop human cloning altogether might be to impose a worldwide ban.
You could make a worldwide ban have teeth if you wanted to, if there was the international will to do that, of course. It's simply not good enough to shrug your shoulders and say you can't do it, and therefore leave that experimentation to take place.
When we asked if nature may take its revenge for experiments like human cloning, most worried are the Taiwanese and the Poles. Least concerned are the Danes.
I don't think that nature will take revenge upon us. I think that nature, that there might be unexpected things happen because it's science, and you might try something and it will not turn out right, and there might be detrimental effects, but that's normal. It's not revenge.
If you treat a human with disease and you make sure that whatever gene you give the patient to help them is not getting into their germline so that it's not given onto their offspring, then I think it's a very controlled and very helpful way to do things.
I think there are dangers that something gets introduced into the society that we didn't plan on, and we just couldn't catch it. We don't really know that there are sufficient safety nets available right now to catch things like that.
Yeah, if we start tampering with genes in the long run, there's probably going to be some damage somewhere along the human race. But I don't think it's going to be anytime immediate, no. But yeah, once we start doing this on a regular basis, if that ever happens, I think the turnaround will get us. So
nature always fights back. And she usually has her way, so that's just how it is, and we're going to learn it with population growth in the world, and if we're not willing to control ourselves, then nature will do it for us.
Every piece of technology comes with a price. There may turn out to be some terrible disaster waiting around the corner, and we can never say no to that. But it seems to me less likely that's going to come from genetics than it comes, let's say, from physics. I'm much more worried about the world ending with an Einsteinian bang than with a Mendelian or genetical whimper. So I think let's get real about the threats of genetics. They're there, but the hopes are much, much greater.
And the promise of those hopes remains extraordinary.
Since the discovery of DNA 50 years ago, I think one of the most exciting developments has been the ability to use this technology to replace various worn-out body tissues. So the hope would be is that within our lifetime we could eliminate diabetes from the planet altogether. Or say, for instance, you got in a car accident and you lost a kidney, we could take a skin cell from you and using this technology grow you up a new kidney.
There will be benefits, but it's also fair to say that we're also going to deliver a lot of harm. So I think what is a responsible approach is to say, look, any new scientific avenue brings with it both benefits and costs, a leap forward and leaps backwards. Our mission is to ask the important question, the Socratic questions, the why questions, the ethical questions, the principle questions up front, so that the journey will be a little bit more beneficial as opposed to being a step backward for humanity.
And it's only by all of us asking these questions and listening to the answers that we can hope to regain control of what many believe will be the age of genetics.
One thing that we can't cope with is disappointment. We can't cope with saying genetics is going to do nothing, let's stop it, or genetics is too dangerous, let's stop it. We have no real option but to continue. And maybe in 50 years from now, I hope, genetics will allow me to come back and look back for the century of the DNA molecule.